Today we're going to start a new series whereby we analyze the images associated with the various laboratories in the histology class. This particular one has to do with images on fundamental tissues and mitosis. It has three parts. One part talked about the types of microscopy, tissue preparation, and staining. The second part has to do with images of light and electron microscopy, and the third part has to do with mitosis itself. If these materials are useful to you, I hope you'll be able to pass this information on to your colleagues and to your friends to have them subscribe to the YouTube site. Thank you. Images of fundamental tissue and mitosis part 2. One of the images we want to see is skin. And we can see skin has different layers. It has a basal layer. So this is the epidermis and the dermis below. And we have the basal layer. We have the stratum spinosum with a bunch of spines, stratum granulosum with granules, and then the stratum corneum. And then in through here, we have mostly collagen fibers, which are extracellular matrix, and then there's fibroblasts. You can see nuclei of fibroblasts are running through there. So this is the epidermis and the dermis. So this is epithelium, and this is connective tissue that we can see, and we can see little blood vessels running through there, maybe a nerve or two. But this is the basal layer, the granulosal layer, and the stratum corneum that we see there. And in the we see swirls. Most of this is collagen, extracellular matrix, but the nuclei that we see here mostly are fibroblasts that we can see. So if we take a look at that, we can see this is the skin. So this is the epidermis. This is stratum corneum, stratum granulosum, as you see the granules in through there. Stratum spinosum, I don't know if you can see the spines between adjacent cells, and the stratum basal. So this is the nucleus, and this is the cytoplasm of these cells. But if we look in the dermis, we can see little capillaries in through here. Maybe that's the arteriole, with the smallest arteriole we see there. You see group bundles of collagen, extracellular matrix, and you see these fibroblasts located in through there. And then these are again, the connective tissue is organized in different arrangements so they can resist stress from whichever. Collagenous material is organized there. Also, we have the sweat glands, and we can see these glands. There's a granular portion here and the duct portion. The duct portion is cube cells on the surface, and it's more than one layer, so it's stratified cuboidal. So this is a duct, is more eosinophilic, another duct in through there. So secretory part of the sweat gland, and the duct part which takes it to the surface, and that's what we're seeing in through there. Here's a little arteriole, or maybe a little small artery or two arteriole. This is nerve. So this is nerve, kind of swirly stuff is nerve. This is another little arteriole or small artery. These are endothelial cells, or red blood cells, and then these are smooth muscle cells. So we see the nuclei and the cytoplasm of these cells. This nerve that we see here, we see connective tissue bound on the outside, but we can see the cells inside, which are myelinated axons that we're seeing there. We don't see the cell bodies per se. Epidermis, dermis. Dermis is connective tissue is dense and it's irregular. Again, we see some nervous tissue in through there, bundles of muscle that we see in through here. Blood vessels, there's an artery that we see, several layers of smooth muscle, flattened endothelial cells around the surface, and then we see some fat cells. So these are fat cells. So this is one fat cell, and there's its nucleus. There's another one, there's its nucleus. There'll be another one right in through there. So these are fat cells that we see. The fat, they say, looks like chicken wire. Is kind of what it looks like to identify. Again, these are blood vessels, arteries that we have there. Sweat gland, sweat duct, blood vessel, nerve that we see in through there. And here, these are actually nerve receptors in through here. This is the Meister's plexus that we learned about. It's a capsule around some nervous tissue to detect touch. And so if we look at the next one, we're supposed to look at tongue, and we can see that this is epithelium on the surface, and then this is connective tissue in through there with a lot of muscle, because there's a lot of muscle in tongue. And then also we can see there's glandular portions. So there's glandular portions that is being produced as well. And we'll see muscle. So we'll see muscle. So if we inside here we see where the muscle is, and the next slide shows you what that muscle is. It's a skeletal muscle, and the nuclei are on the periphery, not in the center, but on the periphery, and it's a multinucleated cell. There's more than one nucleus along the way. Here we see endothelial cells of the capillary of the endomysium. So it's a capillary, and these are the muscle cells, which are composed of more than one nuclei. The muscle cell, this is the endothelial cell that we see here. And so these are muscle cells, skeletal muscle cells, and this is a serous secretion, kind of red secretion through there. These are fat cells, a chicken wire type, and what we're seeing are strips of skeletal muscle that runs through there, and in the very surface up there we'll be able to see the epithelium. So this is connective tissue and muscle, and here we can see the smooth muscle cells with the nuclei on the periphery, here, here, and here. So this is one muscle cell. There's another muscle cell in here, very large cells. And then this is a secretion. This is an actinus. Actinus is like a grape hull of cells that secrete into a lumen. And we can see the spherical nuclei located in the secretion. This is a serous or proteinaceous type secretion. And so if we look at that, we can see a little piece of tongue. This is a little piece of tongue, and we can see little projections on the tongue, and these are filiform papillae. Don't worry about that. Just to say that these is what grip the food to be able to move the food underneath your teeth. So you got epithelium, and then you got connective tissue. Again, we see these pink bundles of collagen, and the dark cells are the fibroblasts. And here we see a little capillary. We can see the endothelial cells of this capillary. The epithelium is stratified squamous epithelium, non-keratinized, because it's in the oral cavity. So we see fat cells, and we see, so these are fat cells in through here, the white ones that we can see. I'm trying to get a good chicken wire shape so you can see this is the fat cell, this is the nucleus of the fat cell. And these cells are the muscle cells. You can see the nuclei on the periphery, and there's multinucleated, and you can see striations that occurs along through here. Skeletal muscle is striated, namely you can see little cross bands of it as you look at it. So you can see the little striations there very, very nicely. Also, we should look at secretion. This is a serous type secretion. It has spherical nuclei, so this is one of those acini. Another acini right there, another acinus right there. Acini are more than one. Acinus is one. Here's an acinus right here, and you can see the nuclei of the cells and the secretory products. And the nucleus is not necessarily on the base, and it's spherical in shape. And that is in contrast to other type of secretory units that we might see here. But there's also serous. So there's serous and mucus, and so what we've been looking at so far, this is the serous ones. Somewhere there should be some mucus ones. I thought the mucus was more numerous. Here we can see the mucus. The mucus one, the nucleus is flattened and it's at the base with this kind of lighter staining material. Serous, mucus secretion. Sometimes they're in the same one. So we're able to see that. This is a blood vessel in through there, and then the secretions go out into the oral cavity. So the next one that we would see will be the kidney. In the kidney, we saw it before, the cortex and the medulla that we'll see. And here we can see the proximal tubule, the distal tubule in through there. And so that's what we're going to see right now. And so if we look at, well, I guess we don't have the kidney to view at this moment. But what we did see is the thyroid. So this we'll take a look at the thyroid. Thyroid has simple cuboidal cells around a follicle. So it has follicles. There's follicles here, here, and here. And you have these groups of cells that make a follicle. Thyroid globulin is what's inside their storage component. In between there you have the parafollicular cells, which produce hormone too. So these cells produce thyroid hormone, but this is thyroid globulin that's inside. And we see that in slide 75. You can see the cuboidal cells here, the thyroid globulin in through there. This is a blood vessel. This is the connective tissue in through there. And so if we look at that, we'll be able to see the thyroid. These are the follicles. There's one here, one there, different sized follicles. And if we take a look at one, you can see these cells here, the kind of cube-shaped cells. You can see the nucleus there, and the cytoplasm is a pink kind of around there. And then this is extracellular colloid that's located here. Thyroid is richly vascularized, like most endocrine cells. You can see the endothelial cells here and here, and the red blood cells inside there. In between the follicles, you see the parafollicular cells that we could see. The next one that we were looking at would be the pancreas. And here we can see the cells of the pancreas as the islets of Langerhans. They produce insulin, but also we're looking at the pancreatic aster cells. So aster cell is in the astnus, and they have like little pyramid type cells that we can see. And here we can see it. This is in the pancreas, and so this is an astnus right here with a series of cells around a secretory unit. Each one of these pancreatic aster cell is pyramid shaped, and so it's astnus, remember, like a grape hole with a secretion in the very center. And so you also may note that the secretions is red, which is protinaceous, remember, eosin staining, and the base of the cytoplasm around the nucleus, there's the nucleus and the euchromatin, heterochromatin inside there, and then the cytoplasm around it is staining blue too. That's because they have a high density of ribosomes that we see there. So if we take a look at the pancreas, that's what we see. This is the islets of Langerhans, but today we're concentrating on an astnus. There's one astnus there. There's another astnus there. You can see the nuclei of these cells with the nucleolus in the center, and you see the blue cytoplasm, which is a high density of rough endoplasmic reticulum, which is what's required to make secretory proteins. And so that's what we see. And if you happen to look at the EMs associated with this lecture, this is the rough endoplasmic reticulum membrane that we see there with ribosomes on the surface there. Again, you can see the ribosome associated with this is a nuclear envelope, and you get ribosomes on the outside there, and also in the granular or rough endoplasmic reticulum. We see more rough endoplasmic reticulum, and this it's the ribosomes in these that are making the cytoplasm blue. So rough endoplasmic reticulum again. Also we see mitochondria. Mitochondria double membrane. Rough ER is single membrane. And then also we can not only have smooth ER, we got rough ER. We got rough ER, we got smooth ER. That is, we have no ribosomes on the agranular one, and these are produced steroids as opposed to rough endoplasmic reticulum produce proteins. If you see an endothelial cell by itself, you might even touch back on itself. As you see there, there's a nucleus, there's the lumen. This is a blood cell, but you can see little vesicles pinching off. Those little penocytic vesicles that pinch off that care fluid from one to another. You see one of those here. And then we can also have mitochondria that we can see in this one. This is the nucleus, the rough endoplasmic reticulum, mitochondria, which is a double membrane mitochondria here, mitochondria there as well. Lysosomes, these secondary lysosomes, as we can see, but we'll see mitochondria. A higher magnification, you see mitochondria with a double membrane, and you see the rough endoplasmic reticulum with this is the space in between where the secreted proteins first appear. Mitochondria, rough ER. This is lipofuscin or secondary lysosome. And so here we see in the liver, we see mitochondria, glycogen, which is a little larger and forms rosettes as opposed to the ribosomes. In the Golgi apparatus, this is more typical Golgi apparatus, as we see there, pancakes with a kind of sickle shape there, rough endoplasmic mitochondria, but the Golgi complex. And here we see a lipid inside a smooth endoplasmic reticulum that we see. What happens is the smooth ER resynthesizes the triglycerides in intestinal absorptive cells. And here we see the brush border, which are uniform microvilli of the same shape. And the microvilli are projected up because they have microfilaments projecting them up. Golgi, which have secretory granules associated with that, and then the lysosome. Again, lipofuscin, again, glycogen, mitochondrion, glycogen, mitochondrion again, and we see the axoneme with the 9 doublets in a central pair. These are ciliate. These are actually they're flagellum of sperm. And then here we see centrioles. Centrioles, chromosomes in through there, microtubules in through pulling these things apart. So cytoskeletal components, you have microfilaments which project up through there, in through there. And here you can see, if you saw a cross-section of this, you can see the microfilaments projecting the surface of the cell, the membrane up. And you can see these projections. So this would be the microvillus, and then you can see this is a plasma membrane, and it turns into the terminal web. There's a web of it, doesn't have that much cytoplasm in through there, because that's where you have all these filaments associated with it. And remember, when we looked at the PAS staining brush border in the kidney, that's what we're seeing. We're seeing the glycocalyx coat, which is on the outside, the fuzzy thing you see on the outside. That's where it has a carbohydrate rich, is what we see there. Thank you.